So before the transcript can leave the nucleus, it has to be altered slightly. And there are a few reasons why this is going to happen. For one thing, we need to give it directions on how to get to where it needs to go. And for another thing, we may need to snip some pieces out of it, pieces that aren't really part of the information we want. So if you're a bacterium, your DNA is just naked DNA. It's not wound up, it's not in chromosomes or whatever, it's just DNA, and it's sitting out there in the cytoplasm already. And so when you transcribe it, as you're transcribing it, as the strand of RNA is growing, it's also being translated into a protein right away. There's no nucleus to worry about. It's very efficient. But bacteria only have a few genes because they only make a few different proteins. So it's okay to do this. When you have a cell that's a eukaryotic cell that may be making hundreds of different types of proteins at the same time, you can't do it this way. You have to compartmentalize and make things a little bit more streamlined. So in humans, your messenger RNA transcripts, right after they're copied, they're modified slightly before they can leave. So the first thing that happens is they put a little cap on the 5' end of the transcript. And the cap is just a sort of part of the instructions that tell the cell where to go, as is the poly A tail. About 100 to 200 adenines are added at the end of the thing, the 3' end, forming a poly A tail. And those both help the messenger RNA get out of the nucleus and find a ribosome. So think of them as instructions on where to go. But that's not the most important processing that happens. What actually happens with your RNA is scientists started to notice when they were able to isolate these things that the length of an RNA transcript often was shorter than the length of the gene it was copied from. And they wanted to know why this is. How is it? If it's a copy, right? Copy should be exactly the same. But it wasn't. It was shorter. Sometimes there were large pieces of information missing from these messenger RNAs, and yet people were surviving and living perfectly happy lives, so it must be normal. What's going on here? It turns out that within a gene in your DNA, there are sections, long sections of bases that aren't actually part of the information to make the protein that that gene codes for. So there are these non-coding areas right in the middle of your gene. It's like sort of reading a sentence, and in the middle of the sentence that you're reading is a recipe for chocolate chip cookies, right in the middle there. It has nothing to do with the sentence you're reading, but it's stuck right in there. Well, if you want to make any sense out of the sentence you're reading, what do you have to do? You've got to take out these pieces that don't have anything to do with it so that the sentence makes sense. These non-coding areas are called introns. The areas that we're interested in, the part of the sentence that you want to read, is called the exons. And the way I remember this for me is X means exo, or X means outside. And so think of it this way. The exons get out of the nucleus. The introns stay in the nucleus. It's a way to help you remember that. So what happens is we've got to get rid of those introns. We've got to take them out of the transcript because they don't contain information that we want copied into protein on the outside. It's not junk, but it's not important to the protein. We don't see these introns in bacteria. We only see them in eukaryotic cells. And the reason for that primarily is that we've had a lot more evolution to create a very complex genome that we have. A lot of hitchhiking pieces of DNA have sort of found their way into our genome. They don't really affect us in any negative way as long as we're able to snip out those pieces before we try to make a protein. Okay? So even though they're not part of the important information in the gene, because of the way transcription happens, they're copied anyway. RNA polymerase doesn't know what it's copying. It just makes a copy of what you tell it to. It's just like a copy machine. A copy machine doesn't edit. It just copies. Same thing with messenger RNA, or with RNA polymerase. It just copies. It doesn't edit. So the unimportant part of the information is still in the transcript, and we need to take it out. It needs to be cut out. There are small nuclear ribonuclear proteins, which we call SNRPs. And SNRPs, their job is to cut out these introns, cut out these pieces of information that aren't important. They're made of ribosomal DNA combined with proteins. And a bunch of them work together and they cut out the introns. And then they also splice the exons together to make what's called mature messenger RNA. The sentence is complete and easy to understand, and that's what's sent out to be translated. So here's an example of a section of DNA. This is a gene, and you'll see there's an exon at the top. That's important information. That carries information to make the protein. Then there's this section of stuff that's not really important, and then there's another exon again. We can look at it this way. Up at the top we have our chromosomal DNA. That's the DNA that we're going to copy. And we copy exactly base for base with RNA polymerase. We get this transcript. Nuclear RNA that's inside the nucleus. Those little cross-hatched regions are parts that aren't important to make the protein. So we need to get rid of them. So SNRPs come along and cut them out and then splice together the exons to make one continuous gene. And that's the messenger RNA that gets out. It goes out to be synthesized into protein. There are some interesting things about these introns. They can be as big as 100,000 bases in length. Exons are usually 100 to 300 nucleotides in length. So you're talking about these long stretches of what we used to call junk DNA in the middle of your gene. Like I said, it's like trying to read a story, and every now and then there's a recipe for chocolate chip cookies, or a laundry list, or a description from Wikipedia or something in the middle of your story. Makes it really hard to read. Now, if you're copying that, if you're RNA polymerase, you're a scribe, you don't care. You just copy everything you see there. But in order to make sense of it, we've got to cut out these pieces of useless information. And they could be very, very big. From gene to gene, the exons vary. And we don't know why. We don't know what causes them to change length or to be different lengths, but every gene has some kind of introns in there, and they're all different lengths, and they're in all different locations, and we don't know why. They're still trying to figure out what they're there for. They have some ideas that maybe introns are part of a way for your cells to stop making proteins even after the gene has already been copied. And we'll talk more about that later. What's also interesting is that it's possible, because these exons are sort of spliced together, it's sometimes possible for you to splice them together in different ways to make completely different proteins. Remember, we talked about there being about 22,000 genes, but over 100,000 different proteins that need to be made in your cells. How do you get 100,000 different proteins if you only have about 22,000 genes? Well, part of the way you can do that is by taking the important genetic information, the exons, and splicing them together in a different order. So you have a gene that can produce maybe 2 or 3 different proteins by just how you put the exons together. There's other ways to do it too, but that's one of the more common theories as to how we have so many different proteins from such a limited number of genes. So it allows us to specialize, and that's a really important thing for a complex organism like a human being. Specialization is crucial to survival. So make sure you take good notes, ask your questions, and we'll talk about the next stage in the next video.